Okay, so our final talk is from Karam, who's joining us from Turkey, Samsung in Turkey. So, Karam, if you can, well, you've shared your screen, and you're going to talk, give us 10 minutes about nematodes and CRISPR. So, off you go. Thanks very much.
Okay, hello everyone. My name is Karam Mostafa. I am postdoctoral researcher at Agricultural Biotechnology Department at Ontokusmais University. Actually, after the valuable lectures we just heard, I will keep my talk brief and focused. Today, I will introduce a new era in combating nematodes through innovations. My aim in this presentation is to highlight how modern strategies and technologies are opening new opportunities to manage nematodes more effectively. Actually, I like this video. As we see here, nematodes are a type of roundworm. It is directly targeting plant roots and form call formation, which disable plant to absorb mineral and water from the soil, and the plant will die. Additionally, nematode infections are always combined by other plant pathogens, such as bacteria and fungi. Nematodes can impact several groups, as we see here, peppers, tomato, potato, sweet potato, watermelon, and carrot, and eggplant also. According to American Society of Phytopathology, the economical losses due to nematode infection have been estimated at 173 billion per year. At the same time, the scientific effort also increased during 1990 to 2020 to reach more than 1,000 articles published in 2020. So all of these are indicators that we are facing a big problem. When we are facing a problems, what we have to do, I think we have to take a nap in the lab. Actually, different approaches are used to manage nematodes in different groups, such as soil fumigation, resistance, nematicide, grafting, group rotations, and cover groupings. But despite all this effort, the problem still persists. For potato, we can consider potato as economical group for different reasons. Potato is the world's third most important food group after maize, rice, and wheat. Also, it is grown on almost every country in the world, and nutritional value of potato. Nematodes represent one of the most destructive problems for potato production for various reasons. As we see here, several different nematode species target potato. At the same time, some kind of these nematodes target both below ground and above ground. Also, their feeding activity impairs root development and ultimately result in fewer and smaller tubers, and also the long-term viability of eggs, which can persist in the soil for 40 years without the host plant or potato plant. CRISPR on potato has been applied for different purposes, such as starch quality, reducing browning, development architectures, herbicide resistance, virus resistance, and disease resistance. But CRISPR specifically for nematode resistance in potato are still lacking in application. For this reason, my colleagues and I wrote this review article as titled CRISPR versus nematode, a new area of genetic solution for potato group protections. In this review, we have highlighted different approaches that can be used through CRISPR to develop potato plant with enhanced resistance nematode. First approach is to applying plant resistance genes through CRISPR. As we see here, favorable traits can be activated and undesirable ones can be knocked out by CRISPR. At the same time, these genes we can identify by transcriptomic analysis or whole genome sequencing of susceptible cultivars and the resistance one, particularly under nematode infected conditions. The second approach is engineering plant resistance through nematode effectors manipulation. As we know, nematodes secrete protein known as effectors, which manipulate host plant responses to promote successful infections. Silencing nematode effectors by using CRISPR is a promising strategy as it results in decrease in parasitic activity. For example, here in potato developed RNA interfaces-based transgenic lines that effectively silenced the 16D10 gene. These transgenic plants exhibit a significant reduction in number of egg mass per plant, ranging from 65 to 95 for several root node nematode. As we see here, it is wild type and this transgenic plant. The third approach also engineering plant with protease inhibitors gene. For example, when potato plant were transformed to express the rice OC delta D86 genes, the transgenic plants showed approximately 60 to 70 resistance compared to wild type. The same gene also was introduced into the eggplant, leading to transgenic lines that exhibited an approximate 78 reduction in root nematode. As we see here also, it is wild type and also transgenic line. Learning from nature, the fourth, I think this is the best for me. Learning from nature is a promising approach for developing nematode resistance. As we see here, the castor tree is naturally resistant for nematodes. Researcher investigated its roots execute composition and found increase the level of palmitic acid and hyaluronic acid. From this observation, we formulated our aim for next study, which we have already applied in tomato. So our hypothesis here is that increasing the expression level of palmitic acid in tomato plant will enhance their resistance to nematode. After in silico analysis and bioinformatic analysis, we identified 4 genes in potato and the 4 genes also in tomato, named FAT-A, FAT-B1, FAT-B2, FAT-B3, and this analysis was already published in the plant genome. After that, these genes, we cloned these genes, 4 genes, under 35S promoter to increase the palmitic acid, to increase the content of palmitic acid in our plant. All these genes was cloned under 35S promoter, constitutive promoter, and also we use BIPKB004. Then we successfully produced different transgenic line as we see different regeneration stages. Then confirming this is transgenic by hygromycin PCR test. Transgenic plant will keep and non-transgenic plant will throw away. After that, we check the expression level of these genes as we see here, different lines and varying from the 10 to reach to 1,000-fold change. At the same time, the palmitic acid content was increased in compared to wild type up to 45%. Then we have to check this transgenic plant to nematode test. As we see here, we make a bores, 3 bores around the plants. After that, we plant were infected 1,000 eggs with micropipette and left it to 2 months. And evolution of tomato transgenic, as we see here, the reduction of reproduction index was varied from 10 to 100. 100 meaning the plant was completely resistant to nematode. As we see here, it is this wild type completely destroyed by a nematode. And if we look at these lines, it was complete FAT-B3 was completely resistant to nematode without any call formation. As we know also, classical gene overexpression techniques suffer from certain drawbacks, such as non-specific location and the limited number of genes. Because of this, we used CRISPR-Cas3 system to target the same genes to overexpression these genes and increase also palmitic acid in plant. Quickly, we also determine the efficiency of the guide RNA also and the CRISPR-Cas3 system by using resogenesis, as we see here. We can, to be sure before transfer these systems to stable plant. And as we see here, the overexpression was increased. It is not like classical overexpression, but was increased up to 70. And this plant nowadays is under evaluation for the nematode test. What I wanted to see here, we can use CRISPR to target different approach to reduce plant for resistant potato plants and to increase the resistance for susceptible potato plant by CRISPRs, not only by CRISPR-Cas9, we can also use CRISPR-Cas3 and CRISPR combo. I need to say thank you for all the author of the papers. Also, thank you to my supervisor and to our teams. Thank you for Plant It and AAB also for this invitation. And thank you also for your attentions.
Brilliant. Thanks so much, Karam. So I forgot to mention, I guess, at the start that Karam was obviously introducing a review paper that he published in Annals of Applied Biology, and it's great that you could follow on with that with a little bit of your original research as well. So that's great to see that you're making some good progress there with those targeting those fat genes. So if anyone has any questions for Karam, please put them into the chat. We've got one here already. Let me just dig it out. From Tina. So she asks about how do you envisage tackling the problem of parasitic nematodes that can't be genetically modified? I guess, do you have any progress in that field? But I guess, well, you can answer, but I think you target the plants, right?
First of all, I wanted to say something. I am not nematologist. I am biotechnologist because this I only focus in plants. Any questions about the plant, I can answer it. But for the nematology, I can't not.
Okay, so we got a question about the plants a little bit here. So this is from Ronell asking, is it possible, do you know if it's possible to reduce or remove the chemical signals produced by the plants which serve as a hatching factors for the potato cyst nematodes? So is there a target you might be able to, something you could target within the potatoes to reduce those signals? Do you know anything about that?
Yes, we can do this by CRISPR Cas9. Also, we can do this by CRISPR-Cas3 also.
Okay, so you know the genes that you would target to reduce the signals, yeah?
I think I mentioned them in the article, but I didn't remember it now.
Okay.
We speak about.
Okay. So I guess we have a bit more of a technical question from Rita here about the PCR that you showed. What do they actually show? You can't differentiate between the bands. Well, you've stopped sharing now, so you can share again. You can give us, yes, a bit of a lesson on reading a gel. We can do that. We have a little time. If you can share again.
Now?
Yep, we can see it.
This one?
I think so, yes. Yeah.
Yes, this one was for the hygromycin test.
Okay.
Hygromycin test. This was for target the integrated genes.
Okay. Okay.
Because there is a different size one. Okay. This was for the specific genes. we integrated in the tomato plants. This was for hygromycin test gene, because of this, all of them is the same.
Okay, that's good. So let me ask you about this experiment just to finish. So you showed that there was increased root biomass in the edited potatoes.
Yes.
So have you grown those yet to see if you have more potatoes, or have you looked at the yield of those plants yet, or is that ongoing?
Yes, we grow it, and sometimes we didn't saw any differentiation between fruits, but we see some differentiation in the leaves and stem.
Okay.
Because increasing the aromatic acid may be affected in the mm, maybe in the architectures of the plant.
Okay. Okay. So that for future experimentation, indeed. Yeah. Okay. Great. All right, so thank you very much, Karim, for that. And well, obviously that unpublished work, I hope you'll be submitting that to Annals of Applied Biology, of course, or Plant Biotechnology Journal. But thank you very much for our three speakers, Sajja, Ryan, and Karim. I think it's been super interesting, a really good start to this monthly series. As I mentioned at the start, our next webinar is on the 14th of October, and we're focusing on plant health, a couple of talks in that area. And as I said, we will email everyone with the details of that, but also when the recordings of these talks are available. I think it's been a really fantastic start to this series. So once again, thanks to our speakers, and thank you everyone for attending, and we'll see you hopefully next time. Okay, thank you very much. Have a good day, everyone.